Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another topic, that is your TLC, that is your thin layer chromatography. So chromatography, first of all we have to know that it is taken from the Greek word, that is chroma and graphic. Chroma means color and graphic means to write. Okay, so chromatography is simply defined as separation of the material from the compound. When we have to separate the material from the compound, we do that chromatography. Suppose that this is a material which is made up of A, B, and C. Now, what we have to do, we have to separate it into A, B, and C. Now, what we do, we perform chromatography for the separation of this material. Now, thin layer chromatography means that we use a slide which are coated with silica gel for the separation purpose. This is known as a thin layer chromatography. Now, in every chromatography, we have a two phase. One is your stationary and the another one is your mobile phase. So, we have to discuss about it. Let's start the lecture. In today's lecture, we are going to discuss about the definition of the thin layer chromatography, explain its principle and working mechanism, describe the material, procedure and visualization method, and also discuss the application, advantages and limitation of the thin layer chromatography. Definition. Now, thin layer chromatography, which is also known as a thin layer chromatography or thin liquid chromatography or TLC. It is a solid liquid adsorption. Now there are two words. One is your absorption and the another one is your adsorption. See, adsorption means that suppose that this is your surface. Now material just stuck on the surface. Okay, material stuck on the surface. It is known as a adsorption. Okay, and when the material mixed to the sample, then it is known as a absorption. Okay, so this solid liquid this TLC is a solid liquid adsorption method, and here we separate the component based on a difference in adsorption affinity. It is quick method, simple method and economical method. Principle of TLC. So we have a two phase. One is your stationary and the another one is your mobile phase. Now the component which have a more affinity towards the stationary phase are stuck on that stationary phase, and the component which have a more affinity with the mobile phase it goes upward to that mobile phase by that capillary action. Okay, then different movement rate causes separation into the distinct spot. Here, stationary phase attract the component to different extent. Mobile phase carry the mixture upward by that capillary action. Suppose that this is your plate and this is your mobile phase. Now, here we have a material. It goes to the upward direction. So it goes at upward direction along with that mobile phase by this capillary action, because solvent move towards the upward direction, then along with this solvent the material will also move at the upward direction. Component of the TLC system. So the first component is your stationary phase. This stationary phase is made up of the silica gel or alumina. Now, stationary phase, suppose that this is your plate. Now this plate is coated with that silica gel. Okay, this is your this is your plate and this is your silica gel. Okay, so what we do, either we prepare a mixture of the silica gel and just pour into the plate, or either we buy the silica plate from the silica coated plate from the market. This is a stationary phase. Next is your mobile phase. Mobile phase is what? It is the organic solvent or the mixture, and this the selection of the organic solvent is according to that sample that we have to separate. Next is your sample. Sample is the mixture which we have to separate. Development chamber, container for the solvent. Okay, that container should be covered from that plate, otherwise it will be evaporate. Detector, visualization method for spot detection. Stationary phase. Stationary phase is usually made up of the silica gel, alumina, or the cellulose. Okay, here what we do, we coated the thin uniform layer on a glass, plastic, or aluminum plate. It must be porous, stable, and adsorptive. Okay, stationary phase should be porous, stable, or the adsorptive. Mobile phase is composed of either one or more organic solvent, and the choice of organic solvent is depend upon the sample, the sample that we have to separate. Okay, so mobile phase is either made up of the one solvent or more than one organic solvent. Choices depend upon the polarity of the analytes. Analytes means that the sample that we have to separate, like hexane, ethyl acetate, chloroform, and methanol. Sample application. Spot is applied by using a capillary tube or micropipette. See, this is your plate which is coated with that silica gel. We draw a line 1 centimeter at the bottom and 1 centimeter at the top of the plate. Then we take a capillary tube or micropipette and just take a spot here. Okay, made a spot of this sample by using that capillary tube or that micropipette. It should be placed about 1.5 to 2 centimeter from the bottom, that I already told you. What we do, we draw a 1 centimeter above the line and 1 centimeter at the bottom of that plate. That spot should be small and compact because it prevents the spreading. We have to spot that the sample should be spotted in a very small amount and in a compact amount, otherwise it will be spreaded. Development of the plate. Plate placed vertically in a chamber. solvent rises by the capillary action. Now we after preparing the plate, now this is our plate, it is prepared like that. Now what we have to do, we put it into the chamber vertically, and the amount of the solvent is just below that line. Okay, solvent pour into the chamber below the line. Then solvent is rises at a upward direction by capillary action, components separated according to the affinity differences. The component which have a more affinity with that stationary phase is just move slow down, and the component which have a more affinity with the mobile phase move at a faster rate. Developing chamber. Developing chamber is should be airtight container with the solvent vapors. Okay, airtight container because the the solvent should not be evaporate. Solvent should not be evaporate. We have to focus on that. Ensure that saturation and uniform solvent movement. Common type, that is glass jar, beaker with the lids. Visualization of the spot after development, plate is dry. See, when the spot are developed, okay, then what we do, we dry that plate inside the hot air oven. Then after that, we use a UV light, iodine vapors, or the reagent like the ninhydrin for the visualization of that spot. Okay, the practical video will be uploaded in our account. You can see that video for better understanding. Then retention factors is what it is also known as a RF value. It is defined as distance traveled by the compound divided by the distance traveled by the solvent. RF factors is characterized or characteristic of each compound used for the identification and comparison. It is used for the identification and the comparison. Factor affecting the RF value, type and polarity of the stationary and the mobile phase, chamber saturation, temperature, sample size, and coating thickness. Type of the TLC: normal phase TLC, reverse phase TLC, and two dimension. So TLC is divided into into three categories. That is normal phase TLC, reverse phase TLC, and two dimensional TLC. Step in TLC procedure. The first one is your preparation of the plate. Preparation of the plate means that we prepare a silica gel, then we pour it into the plate, then we dry that plate overnight. Okay, this is known as a preparation of the plate. Then spot the sample on the plate. This one. Then develop the plate. Then dry and visualization and measure the RF value. Okay, the detailed procedure will be discussed in the next lecture, in which we see that how we perform the TLC method. Next is your example. Separation of the plant pigment. Extract is taken from the spinach is applied. Solvent that we use is hexane acetone. Means that we use the mixture of hexane and acetone at 7:3. Means that if we take 7 ml of hexane, then we take 3 ml of acetone. Bands: chlorophyll A, chlorophyll B, xanthophyll, and carotenoid. So we get the four bands of that mixture, that is chlorophyll A, chlorophyll B, and xanthophyll and carotene. Application in food analysis. It is used for food dye, preservatives, amino acid, detect the adulterant and contaminant, analyzes the lipids, vitamins, and the flavor of the food product. Application in biochemistry. It is used for the separation of the amino acid, nucleotide, and sugar. It also monitors the enzyme reaction and metabolites applied in clinical and pharmaceutical research. Advantages of the TLC. It is fast and inexpensive, require small sample volume, simple equipment and minimal training, can analyze the multiple sample at once. So when we perform the TLC, then this TLC is very fast and inexpensive, and it also required a very small sample volume. Simple equipment is required. We required only TLC chamber, TLC plate, that sort. Then can analyze the multiple sample at once. Limitation of TLC: less quantitative accuracy, limited resolution for the complex mixture, RF value affected by the condition, manual interpretation error. Comparison with that paper chromatography. TLC uses the silica gel or alumina. In chromatography, paper chromatography, we uses the cellulose fiber. TLC is faster. Paper chromatography is slower. TLC have higher resolution power. Paper chromatography have lower resolution power. Preparative of that TLC used for the isolation and purification method. Thicker layer used, desire band, scrub and extracted. Modern modification HPTLC, it is used for the separation of the fine particle layer for high resolution power. Automated TLC is used for the analyzing the automated TLC analyzer improve the report, re- reproducibility. Precaution: handle the plate carefully, use the clean glassware, maintain the constant condition, avoid the overloading sample. Quantitative TLC like uh, then dense geometric scanning measure intensity, enables the quantity estimation used in drug sterilization and quality control. Interpretation of result: compare the RF value with that standards. Similar RF factors with the same compound. Spot intensity, concentration. TLC versus HPLC. TLC have low cost, HPLC have high cost. TLC should be done by manual, HPLC should be done by automated. They are semi quantitative and fast, and HPLC is quantitative and take a moderate time. Safety measure: use the solvent in ventilated area, wear the gloves and goggles, handle the UV light very carefully, dispose that waste material in a proper way. Summary: TLC is simple. TLC is simple, efficient, and economical, based on a differential absorption, useful for that qualitative and putative analysis. Conclusion: TLC is essential in analytical chemistry, widely used in food, pharmaceutical, and biochemical lab. HPTLC offer the improved precision and reliability. So this is all about the introduction of the TLC, that is thin layer chromatography. I hope you will understand, but if you have any query related to this, you are freely to ask me in a comment section, or for a further detail you can follow the ITLS Academy on different platform, that is Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450, and visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.